Have you ever wondered about the stress that DNA undergoes during replication? It's a fascinating concept, this DNA replication stress. You see, DNA replication is no simple affair. It's a beautifully intricate process that can put quite a strain on the DNA genome itself. Imagine a tightrope walker balancing herself high above the ground, each step a careful calculation. That's what DNA replication is like. But sometimes the tightrope walker stumbles, she falters. That's when we see the appearance of stalled replication and the formation of stalled replication forks. It's not just one thing that can trip up our tightrope walker. There are many contributing factors, from unusual DNA structures and mismatched ribonucleotides to the tension created by the simultaneous processes of replication and transcription. Even the scarcity of essential replication factors can add to the stress. Understanding these stresses is the first step towards understanding the intricacies of DNA replication. So, what exactly causes this stress during DNA replication? Let's dive into the nitty-gritty. DNA replication is a complex process, and it doesn't always go smoothly. There are several events contributing to the stresses that can stall replication and lead to the formation of what we call stalled replication forks. First off, we have unusual DNA structures. These are regions where the DNA does not adopt the familiar double helix structure. These unusual structures can cause the replication machinery to pause or stall, creating stress. Next, we have mismatched ribonucleotides. Sometimes, ribonucleotides, which are the building blocks of RNA, get mistakenly incorporated into the DNA during replication. This mismatch can cause the replication machinery to stall, leading to stress. Then, we have tensions arising from concurrent replication and transcription mechanisms. Imagine trying to read a book while someone else is writing in it. That's essentially what's happening when replication and transcription occur at the same time on the same DNA molecule. This concurrent activity can cause conflicts and lead to replication stress. Another stressor is the inadequate availability of important replication factors. These are the proteins that help the replication machinery do its job. If there aren't enough of these factors available, replication can stall, leading to stress. Fragile sites on the replicating DNA strand can also contribute to replication stress. These are regions of the DNA that are particularly prone to breaking or forming unusual structures during replication. Overexpression or constitutive activation of oncogenes is another factor. Oncogenes are genes that have the potential to cause cancer. When they are overexpressed or constantly activated, they can disrupt the normal regulation of DNA replication and cause stress. Lastly, we have inaccessible chromatins. Chromatins are the material that make up chromosomes, including DNA and proteins. If the replication machinery can't access the chromatin, it can't replicate the DNA, leading to stress. These stressors are part of the complex dance of DNA replication. But nature has its ways of dealing with stress, doesn't it? Indeed, it does, especially when it comes to DNA replication stress. This is where our unsung heroes, the kinase regulatory proteins such as ATM, short for ATM serine threonine kinase, and ATP, or adenosine triphosphate, come into play. These proteins are like the emergency services of the cellular world. When the DNA replication process is under stress, these proteins get the call. They are recruited and activated by DNA damages. Imagine a city in chaos, and these proteins are the superheroes swooping in to restore order. They rush to the scene of the stalled replication forks, stabilizing them and preventing collapse. By doing so, they alleviate replication stress, ensuring that the process can continue smoothly. Without these proteins, our cells would be in a constant state of disorder, unable to replicate DNA efficiently. These proteins play a crucial role in maintaining the balance during DNA replication. What happens when the stress becomes too much and the replication forks collapse? That's a question we can't ignore. When DNA encounters too much stress, the replication forks may stall and eventually collapse, but DNA isn't left helpless. repairing mechanisms spring into action, aiming to reassemble the broken replication fork. This reassembly process is not a simple task. It requires the orchestration of various molecular players and the right conditions. The goal: to mend the damaged ends of DNA, enabling the replication process to continue smoothly. Imagine this as a construction site. When a structure fails, a team of skilled workers rushes in, assessing the damage and initiating repairs. They work tirelessly, ensuring that the structure is safe and functional once again. Similarly, in the microscopic world of DNA, these repairing mechanisms are the unsung heroes, working behind the scenes to maintain the integrity of our genetic material. In the world of DNA, stress is managed and repair is always possible.